All right, so welcome to my show, or my slideshow. We've got Patrick and Gracie and Mally all listening, so it'll be fun to share this with you, Dad. I'm going to do a practice round of this talk with you, and then we'll see, help me get some of the rough edges off. So here we go. All right. So thank you very much for having me here. I'm really excited to be able to tell you a little bit about the work that we've been doing in our lab on a basic helix loop helix gene that we've modified and we've found can upregulate the expression of many different types of dopamine neuron-related genes. It leads to some really interesting questions that are germane to what your interests are in terms of being able to convert human embryonic stem cells into useful and therapeutic types of cells that could be amenable to cell replacement therapy or for modeling disease and so forth. So so thank you, Mike, Jack, Lewis, for inviting me out here, and I'm really glad to be able to share this with you. So with no further ado, let me kind of dig into this. So what we're interested in my lab is a couple different how stem cells actually differentiate and some of the genes that are important for it. And this particular type of project is focusing on genes that help drive cells to give rise to dopamine neurons. So as you know, dopamine cells they're present within the substantia nigra in the midbrain, and they project up to the striatum to help supply dopamine to the brain and help enhance motor function. And so what we're looking at here in the arrow is basically how there's this dark stained here is basically the presence of these neuromelanin-stained dopamine neurons that we typically see in the normal patient and that are lost in Parkinson's disease. So if we can replace those cells, that would be really great. So they have cell replacement therapy approaches in which we're actually taking agents of some sort, being able to actually reprogram cell types to be able to then transfer into and implant into brains where the destination of those substantia nigral neurons are as they project up from the midbrain to some of the deep nuclei within the brain. So we're really interested in being able to see if some of our work can actually connect to that. There's another type of potential benefit in terms of understanding these types of genes that give rise to dopamine neurons is that we could potentially be able to help reprogram cells within the brain that reside there, perhaps have them release dopamine. So there could be resident glia or neurons that are GABAergic or something. Perhaps we can change the way they work. And so there's some preclinical studies that have shown that it's possible with other types of neurons, not necessarily dopamine neurons, but seems like something that we may actually be able to look at participating in as well, or having some kind of contribution towards. So these are the types of questions that we're hopeful that this actually can apply to. Where we started from is basically that my lab is really interested in understanding how genes can actually regulate neural stem cell behavior. And so what we do is typically we can change gene expression in cells and in embryos, and then monitor the expression of different types of markers for changes in cell fate. So we do this in vivo and in vitro. So this first image here is this basically showing how we can actually inject DNA into the lumen of the developing neural tube, and then as the embryo apply electroporation and then monitor those cells over time to see how they actually take on this expression of our gene of interest here. So this is a bicistronic GFP vector that is expressing our NATO3 gene, actually, that I hinted at before. And here's the eye. So we're able to track and see where this is and see what we can, our transfection efficiency, and also do anatomical and other types of analyses to characterize it. So great, that's wonderful. We can also do work in cells, pretty standard type of work in terms of looking at different types of cells to characterize how these genes may actually work, or how different stem cells can actually respond to it. But I want to focus on this in vivo model because this is where the NATO3 question arose from. So one of the things that we were interested in this model, as you can see, is that we have the developing embryo here really early on. How it actually works is that as this nervous system develops, the three layers that form the neural tube so here what we have is the developing nervous system of the chick. This is at about the 10-somite stage. And so before this formation of the neural tube happened, there are three germ layers that are present. The ectodermal layer is the one that actually gives rise to neurons, and is the one that actually invaginates and folds and creates this neural tube here. And the cool thing about that is that you have basically a heterogeneous population of stem cells present here, or neural progenitors here, that can help you be able to characterize how a particular type of gene, whether the absence of it or the overexpression of it, can influence the way that they behave. So that's kind of our model system. And we were interested in taking this model system and applying it to the question about this one gene called NATO3. So this basic helix gene is interesting because it's expressed where dopamine neurons are born. It's also expressed in some other regions, but that's one of the regions that was actually important for it. Hello. Okay. So what we're looking at is that NATO3 is here is basically, it's mapped onto the MyoD crystal structure. We can see how it can bind to DNA with an E protein binding partner. So some of the biochemistry that we know is that it can bind to E proteins and bind to DNA. The site hasn't been characterized, but we can see some mobility shifts with it. And what we can find is, that it's expressed within this region here. So others have characterized it in mouse. We characterized it in chick to look to see and confirm that NATO3 is actually expressed in this region. And so this area that's known as the floor plate, if we kind of look at the smaller image here, if we peeled off the top of this domain right here and looked down on it, we would see the floor, if you will, the ventral part of this of the developing nervous system here. And so by peeling this back, what we're looking at is basically the expression of a marker for the floor plate known as sonic hedgehog, and then this other one, NATO3 expression. You can see basically that this NATO3 gene is expressed in the same region as sonic hedgehog, which is a classic floor plate marker for the developing nervous system. So dopamine neurons actually arise from this floor plate area, and so just as a means to try to help characterize and give you some orientation for some of the data that we'll be looking at, I'm going to kind of go through some illustrations and schematics about how this actually works. So at around like E3 in chick or E10 in mice or whatever, we have this kind of progenitor domain known as a floor plate domain. Relatively little neurogenesis is actually undergone yet. And so we're kind of looking at the schematic is kind of showing this ventral part of the midbrain. The dorsal part, not represented, would be kind of emerging up here, and then there's this open space here that would ultimately become part of the ventricular system. So here we have the floor plate cells here. These will give rise to dopamine neurons as differentiation occurs. So what happens is that basically as development proceeds, there's an expansion of the floor plate cell population. They start to differentiate into dopamine neurons and then ultimately migrate laterally out into their different positions, right, Patrick? Yes. Within here. And so this embryo that's kind of that's shown here is basically kind of showing the different types of regions in midbrain that you can kind of see this kind of setup and where the dopamine neurons can typically arise. Now, dopamine neurons arise within the midbrain. They don't arise. They don't come in, or they're not born in other areas of the central nervous system. So. This is a cross-section basically helping to illustrate that this NATO3 expression is actually within that floor plate domain at E3 and then also at later stages here. And this is placing NATO3 and the two later markers, much like we saw in that earlier schematic, where dopamine neurons, in this case we're using the marker Neur1, are actually present. So what this helps us to kind of see is an anatomical perspective in which we're actually looking at how floor plate cells can differentiate. And in the midbrain, they can turn into to neurons. They can also, in the spinal cord, and then if they don't actually turn into dopamine neurons in the midbrain, adopt a mature floor plate cell fate. So this is what actually happens in the spinal cord. No dopamine neurons are actually born in the spinal cord. They're only the floor plate cells only just mature. They don't turn in. They're non-neurogenic at all, altogether. And then what we have here in the midbrain is this sort of schematic that I was describing to you before. Okay, so fine. What would happen then if we could actually overexpress this NATO3? Can we test and see whether NATO3 is actually sufficient to drive dopamine neurogenesis? This is a little bit of a dicey type of question, but still something that could be important. It's been shown that NATO3 is necessary for dopamine neurogenesis, but could it be sufficient to do so? And we can check and screen multiple different areas of the nervous system to test this. And so the students that we have here are the ones who actually participated on this. And they overexpressed using that model system that I described to you, the chick, basically to see whether they would actually see more dopamine neurons. And so kind of showing our little map that we have here, a floor plate cell would become a neuron, maybe at the exclusion of becoming another floor plate cell or mature floor plate cell or another cell type over here. So can we kind of drive this, this type of fate out of these cells? So yes. One of the things that you'll see and that we'll talk a lot about as we go along is that these cells are important in these neurons undergo maturation, and we can track and see how that maturation proceeds with these different types of markers. So we have a spinal cord floor plate cell here that expresses sonic hedgehog, NATO3, and FOXA2. And then the midbrain floor plate cells, and these are the ones that actually express FOXA2. And then as they start to mature into dopamine neurons, then what will happen is that they'll start to express some of these other types of markers, LMX1A, MSX1, INCREAL, Neur1, PITX3, tyrosine hydroxylase, are some of the ones that you'll be hearing about. So, so this kind of gives you a sense of kind of the maturation. We can kind of summarize that in a bit of a checklist, and you'll be seeing this checklist in terms of how it actually works within the chick midbrain and the chick spinal cord. And then we'll actually show some data, just how it works in this midbrain cell line, and then also human embryonic stem cells, and whether our NATO3 gene is actually important for kind of driving this type of effect. So the NATO3, wild-type NATO3 in this case, is being described here. Can it drive this sort of marker of and make dopamine neurons in areas where they typically aren't made? So when we overexpress the NATO3 gene in the side that's actually treated, the overexpression side here, we can see that there's a greater number of Neur1 positive cells that are present. If you remember, Neur1 is one of those markers which is present at the later stages of once you've become a post-mitotic dopamine neuron, so right here. So, and so what we're seeing then is that basically there's this induction of this type of marker here. On the untreated side, same anatomical section, same embryo. What we see is that on the untreated side, there's fewer of these neurons here. And if we look at, just to confirm whether there's co-expression in these Neur1 positive cells, you can see that, that the GFP expression is relegated to this Neur1 positive population here. And so what I want to kind of point out to you, though, is that this region that we're looking at is right here in the sort of posterior portion of this midbrain. So the posterior ventral midbrain is the area that we're looking at. So we were pretty excited about seeing this, and we thought, oh, great, then now we've uncovered something that's going to make dopamine neurons in a lot of different places, and we've counted them up and found that we see that this is an effect that we see within that particular region really robustly. But when we actually looked in the anterior part of the ventral midbrain, the effect was lost. And so you can see there's Neur1 positive cells expressed on both sides here, but the Neur1 overexpression didn't seem to actually correlate with Neur1 expression. So this isn't sufficient to actually drive these presence of these dopamine neurons. Here you can see that the quantification illustrates that. So, So what's happening in this posterior region, and what are some of the other types of things that can arise? So I won't go through all of the data, but just to summarize, basically, is that we have these types of markers that are associated with early on in development, and then later in development. So we're going from immature floor plate-like cells at Sonic Hedgehog, FOXA2, etc. And then what we're seeing is basically that there's an increase of about 20% in the midbrain of the chick in terms of what the expression of Sonic Hedgehog is for what I call double positive cells, meaning that we've overexpressed our NATO3, and then we're screening to see whether there's actually our markers actually being like Sonic Hedgehog is being expressed there. So, so there's an increase of 20% or so with Sonic Hedgehog, FOXA2. ALMX1B is more robust of about 45%, and then Neur1, 63% increase there. So, we're seeing some of this profile increase within this particular region of this posterior part of the midbrain. And it seems anatomically restricted. So we don't see it happen within the spinal cord here, except for LMX1B, which is an interesting exception. So it may be that the progenitors there are tuned in to being able to respond to NATO3, or perhaps that NATO3 is really proximal to driving LMX1B. Also, these later markers, PITX3 and tyrosine hydroxylase, we weren't able to observe expression by immunofluorescence, but looking for mRNA expression, we found that there is an induction of their expression as well. And that could be just a limitation in terms of how late we could allow the process and the amount of time that they could actually develop in our system. So, okay, so it looks like we have NATO3 actually driving dopamine neurogenesis in a really specific type of area, only in this ventral posterior midbrain. But that's not really useful for us to making dopamine neurons. And so our idea basically was like, well, maybe there's something else going on that makes it so that this area of the nervous system is really special and is responsive to it. And so maybe there are, factor X could be somewhere here or right here, but not here and here. And this would actually make sense because NATO3 is expressed more broadly than just in that ventral or posterior midbrain. And so it really could be something that's, useful for, or it could be that there's some other, necessary cofactor that's required. So Dan Doyle, among others, and are students that were involved in my lab in trying to examine this type of question. And so we went through a couple of different types of options here. So one of them is basically that, what is this factor X? And one is that we have genes that are enriched in dopamine neuron stem cells. So we tried a couple of different genes, like OTX2, FOXA2, and LMX1A. But we didn't really see any kind of co-expression of NATO3 with them, didn't seem to really kind of have that effect of expanding the domain in which we could actually see dopamine neurogenesis. And we tried a number of other things like ASCL1 and neurogenin2 that could be co-expressing with it, and combinations of these different types of genes. And then trying it in midbrain cell culture with a special cell line that we have that we obtained to see whether that might actually sort of unlock the potential of wild-type NATO3. And have it expressed more broadly. And we really just didn't see that much of an impact on it. But we had one hypothesis that was a bit far-fetched. And so this is sort of what would happen if the gene was actually somehow phosphorylated? And it's kind of silly in some ways because it was such a far-fetched hypothesis, but it was relatively easy to test. And so that was what led us to generating and modifying the NATO3 gene to see whether actually mimicking phosphorylation would actually be could enhance this ability to drive dopamine neuron marker expression. So if we look at some sites of potential phosphorylation, there are some that are actually really interesting. It's T132. There's also another one that's not shown here, T101. And then also serine 140. And so these different amino acids are being shown in their conservation. They showed up as being substrates for either casein kinase or PKA, cyclic AMP-dependent protein kinase. Some other common kinases, AKT kinase as well, in their regulation or as far as capacity to be able potentially to be regulated. And we were curious about that. So these are basically showing the different types of genes, the NATO3 genes here, Drosophila, Chick, mouse, human, and so forth. And then these are all of the different basic helix of helix genes that share significant homology with it. And you can see that these threonine sites here and serine sites here are really well conserved. And what's really interesting with this S140 site actually is that it's well conserved in ASC, or it's been shown to be critical in the function of ASCL1 and also OLIG2 in terms of how those BHLHs function. So we were curious as well, and went ahead and started to characterize and started doing modifications of them. So showing on this crystal structure, basically we're looking at this within this loop domain here, where we have this, where we have these two different sites here. And so I've called this phosphomimetic 1, and then this one's called PM1 and PM2. And what we did is that we found, for a longer story, that if we also modify the threonine 101, that along with these, that we seem to see a much more robust type of effect. So PM1 is this one that's where we've actually overexpressed this this modified NATO3 gene. This is the treated side here of the developing midbrain, and then this is the untreated side. And this is a marker, Sonic Hedgehog, which is a floor plate marker. And if you remember, Sonic Hedgehog is one of those really early markers here. Its endogenous expression is right here along the floor plate. And on the treated side, we're seeing a lot of different broad expression of Sonic Hedgehog here, where you don't see it at all on the untreated side. So it's a really profound effect that we see here. And so if we kind of zoom in on that domain and look at it, we can see that there's co-labeling of where we have this ectopic expression of PM1, and that most of this looks like it's, we don't see any kind of Sonic Hedgehog being expressed in cells outside of the ones that are actually PM1 positive. So the ectopic expression that we see of Sonic Hedgehog seems to be solely dependent upon the overexpression of this modified gene. So this is really interesting. And if we look at other types of genes for dopamine progenitors, for example, LMX1B, what we find is that that we have expression of LMX1B pretty broadly within this domain as well. And it's not the same percentage that we saw here with Sonic Hedgehog, but we still see that there's a dramatic number of these cells that are actually positive. So, so Sonic Hedgehog and LMX1B are positive. This seems to, maybe we're on the right path, and we found this type of modified gene that can actually be really critical for helping to drive dopamine neurogenesis. But when we looked at we also found that it really did this very broadly within the spinal cord. So this is a depiction of the spinal cord. This is the untreated side, and this is the treated side. And on this treated side here, you can see that we have a broad expression of this marker here. Hi. Yeah, I know. Oh, you're helping, aren't you? So this marker here, FOXA2, is a floor plate marker, and we can see its expression right here normally, and then its expression is broadened. And actually that we see the expression of these GFP positive cells all the way out here in the dorsal root ganglion. So this is really crazy. I've never seen anything like this with the wild-type NATO3. Clearly something really different is happening with this modified gene that we've been characterizing. So what we did then is what we wanted to look to see whether we actually saw dopamine neurons. This would be great. We didn't see evidence that this PM1 gene actually helped to drive mature markers for dopamine neurons, or markers for dopamine neurons that are post-mitotic, like Neur1, for example, or tyrosine hydroxylase for that matter. So the treated side is over here, and the untreated side here, and we really just don't see an expansion in those numbers. That's interesting, especially considering that the wild type tended to do this type of effect, but that wild type effect was very focused, and so and it may be that this type of modification doesn't necessarily lend itself to the Neur1 type of expression. So really interesting in terms of how it has this really robust expression. And I should remind you that if you remember in the wild type for the for this NATO3, we saw a very subtle type of induction of Sonic Hedgehog here. Nothing along the lines of what we're seeing here, where 100% of the cells are Sonic Hedgehog positive. So what it seems like is that this PM1 version, this first version of phosphomimetic gene, is important for helping to promote this sort of floor plate progenitor fate that we have. And it may be there's more work to be done to help sort of clarify this, but what we're looking at basically is that perhaps we're actually driving the floor plate fate and permitting it to actually replicate, and that we aren't necessarily differentiating into dopamine neurons themselves. So what about this other modification that we talked about, the serine 140, and we made the double mutant, the T101E S140D, to characterize that. And here what we're looking at was basically to see what the effects were. This was actually really exciting because it acted a lot like PM1 in terms of driving those expression of those early markers, but it also helped to drive the expression of some of the later markers that you see in dopamine neurogenesis. So if we look here, for example, FOXA2 expression, this is a floor plate marker, really broadly expressed over here. And in this is really interesting. This is the anterior portion of the midbrain where wild-type NATA3 didn't work before. And so in this region, and actually even in regions of the telencephalon, anywhere that was rostral to where we were of the midbrain-hindbrain boundary, showed this type of effect of these early markers like FOXA2 being induced, and also NER1 being broadly induced in regions where we typically didn't even see it before. So this is far enough in the anterior midbrain and getting into the diencephalon where we just don't see these NER1 positive cells arise. And so this is really kind of an exciting type of approach, because what it suggests is that we've really expanded how NATA3 works by making this modification at the S140 and T101 sites. So other markers, and GRAIL1 is also upregulated in this type of context as well. So this led us to being really excited that this type of gene could be really important for helping to drive dopamine neurogenesis. And we looked at to see whether this could actually encourage dopamine, these types of markers in other cell types besides just the chick. Are we just like dealing with something that's weird with the chicken, or are we actually seeing something that can that we can characterize a little bit more in a cell line? And so the cell line doesn't replicate all aspects of what we saw in the chick, but we do see some of these different types of markers like sonic hedgehog, LMX1A, and NER1 being upregulated with the phosphomimetic. This is the PM2 phosphomimetic NATA3 or PM NATA3 here. So the wild type is being shown here, where we do see some induction in that cell line of LMX1A, of NER1, and so on. So exciting and interesting. So what we want to do is to try to basically summarize what we have, where we're looking at how the PM1 is helping to drive some of these early markers here, but doesn't seem to necessarily have enough to help push the ectopic expression of NER1 later in development. PM2 seems to help drive this later series of markers, including in GRAIL1 and NER1. We didn't see expression of tyrosine hydroxylase at the stage that we were actually able to assay within the chick, and we didn't see it in the midbrain cell line as well. But more work to be done there to see whether we can actually bring out some of that might just need a little bit more time to identify. Still, we wanted to be able to see, like, well, maybe this is something that we can actually use to help generate dopamine neurons from human embryonic stem cells. And so I worked with Mike on generating using the systems that you guys have developed in terms of the modification of the Sudoff approach, where we can actually be able to drive the proneural gene neurogenin 2 along with our phosphomimetic NATA3. And basically the idea is we wanted to see whether we can actually help just push and create dopamine neurons, or at least any of these marker expressions within human embryonic stem cells, and track and see what happens in the H9. So we're basically comparing two different cell lines. One is the neurogenin 2 by itself, and then the other one's the neurogenin 2 with the NATA3. And then these are under the doxycycline inducible versions of it. So what we're looking at basically is that we plate the cells in E8 medium the day before we actually add the doxycycline. And then 2 days after adding the dox, we basically pass into the 3N media and then remove it on day 9. So that's the area that we're looking at, and then we allow them to differentiate. So if we kind of look at what we're, the expression of the types of genes that we have here. So when we started pulling the data out, what we find is that there's that when we added the doxycycline, we also found that there was an induction of our neurogenin line. So the red line here is representing the PM-NATO3 with neurogenin. And then the blue line is representing our control, which is the neurogenin 2 only line. Yes. Yes. That's right. Oh. Well, you're going to have to hold your question until the end. Okay. All right. So so this basically shows that our induction worked in terms of driving the expression of neurogenin. So we're tracking neurogenin expression in this type of mRNA expression. So we use qPCR on harvested cells at these multiple different time points to characterize the gene expression of them. And so PM-NATO3 also was induced, and it was induced in the cell line that is carrying both the doxycycline-driven neurogenin 2 and the doxycycline-driven PM-NATO3. So here we see NATO3 being upregulated in the presence of doxycycline. So great, awesome. So now what we want to do is we want to basically look at and characterize and see what happens to other types of genes that are related to dopamine neurogenesis. So one of the things we're looking at is say, well, do floor plate markers, those early ones that we see, like sonic hedgehog, do they upregulate? What about FOXA2? So, so those markers, we didn't really see any changes in FOXA2. We actually had a hard time actually detecting FOXA2 after day 0. So this we tried 2 different types of primers for it. I think that we have just lost FOXA2 in this cell type. We did see an induction of sonic hedgehog, however, in this type of in this condition. So it seems that after a doxycycline type of treatment here, afterwards, as the cells were allowed to differentiate, what we found is that there was an upregulation of sonic hedgehog here. So, it's a modest type of expression here. We're looking at about, a 4-fold change or so here, but still something that could potentially be meaningful. The other marker, if you remember, we looked at INGRAILD1, and PM-NATO3 was really powerful for being able to drive INGRAILD1. But we don't see that type, and we see some of that expression happening here, where this is day 0 here. When we add the doxycycline, we see this drop in the expression of INGRAILD1. When we release the cells from doxycycline, then what we see is that we see this uptick in INGRAILD1 expression that is unique to what we have here. So I have to say, to qualify this, that this is an N of 1. So these are preliminary data that we're looking at in terms of seeing how these different markers actually are induced. But this is where we had to kind of start with, and part where it'll be interesting to get your feedback. So what about these other dopamine neuron markers? Were any of these actually upregulated here? And we looked at Neur1 and tyrosine hydroxylase, and we actually saw that there was a nice uptick of Neur1 after the release of doxycycline. We can see that it's expressed here. It's about 35 to 37-fold here, and here it's at about maybe 10-fold relative to what it was beforehand. There's an interesting drop-off here at the last date that we actually harvested here that I'm not sure what to make of. Also, interestingly, the tyrosine hydroxylase expression was really robustly expressed in the embryonic stem cells as they were allowed to continue differentiation. And so what we have here then is that we have this induction here of, this is about 25-fold here, for example, and then this is about 250-fold. So there's like a 10-fold difference between these two here. And then here, this is over 1,000-fold difference here and fivefold here. So it's really a dramatic difference in tyrosine hydroxylase expression in that condition. So if we want to, I'm going to skip this slide and delete it. And I'll delete this one. So if we want to look at how these genes were working and how they actually behaved in these different cell types, then what we find is we can summarize it in the following here. So I basically put pluses instead of the numbers that I had before just to try to keep things simple. But we're looking at basically that, Sonic Hedgehog is upregulated in a lot of these different cell types, and we see an uptick in the human embryonic stem cells. I'll put a number in there. FOXA2, we see this upregulation in a lot of the different cell lines. We don't see it in the human embryonic stem cells that were treated with Neurogenin-2 and PMNATO3. LMX1A, it was difficult to characterize the LMX1A expression in the chick, but in the mouse midbrain cell line, we did see an induction of LMX1A with a PMNATO3. We did not see it in these human embryonic stem cells. Likewise with LMX1B, which was so robust in the chick before. And then we look at what we see for these other markers, and I think the one that's the most interesting is that tyrosine hydroxylase between 10 and 200-fold induction relative to the Neurogenin-2 alone. And then the Neur1 that has that 3.6-fold increase but then drops off. So some of these markers are moving along. But bona fide dopamine neurons are usually FOXA2 positive, Neur1 positive, TH positive, and express robust amounts of those. So there's a lot more to be done. These cells, we need to be able to go through and look at immunofluorescence and characterize some of the behavior that way. We need to repeat the experiments again to ensure that we're actually seeing the same types of expression patterns that we saw the first time. And to get some insight into how these genes might actually be regulated by this phosphomimetic NATO3. But I think a larger question is, we want to be able to kind of open this up. These are not looking like real dopamine neurons. Is there a way that we can kind of explore this a little bit more thoroughly? So the way forward, if you will, in what I'm thinking about is that there might be other factors that might be needed to help drive this dopamine neuron expression. So there's a 20 to 31 day protocol that basically that's a standard one that's in the field and we can use to generate dopamine neurons. And so we can track NATO3 expression typically in this typical method. We use that method, the SUDOC method, which is kind of accelerating dopamine neurogenesis. But if we use the one that's human embryonic stem cells and adding multiple different factors to help create dopamine neurons, then that might help us to be able to track dopamine neuron expression. So just to give you a picture of that, the standard protocol is along the lines of where we actually create floor plate cells from human embryonic stem cells here. And using a dual SMAD inhibition, which you guys are familiar with. And then in addition to Sonic Hedgehog agonists and GSK3 antagonists to help mimic Wnt activation, some sort of midbrain sort of specific types of events that are occurring. And then this sort of maturation survival step here. And so what we're looking at then is adding things like FGF8, BDNF, ascorbic acid, etc. And so usually what happens is that we see the FOXA2, OTX2, LMX1A kicking on in around day 13, and then around day 20, TH and Neur1. So we're going to take a commercial version of this type of protocol and then monitor to see where NATO3 might be induced just under normal conditions. And additionally, take a cell line that actually is expressing PMNATO3 only without Neurogenin-2, and then seeing if we can use that to help goose this protocol into differentiation. So what I'm hoping or thinking is that perhaps we can be able to create many different types of floor plate progenitors, which is relatively easy to do, and then be able to add this or activate this PMNATO3 to be able to help push them into to maturation quickly and easily, perhaps much more speed than what we see in the typical cell culture conditions. So that's kind of the approach that we're looking at there. So now you've gotten kind of a picture of what we've been looking at in terms of how these genes actually work. I think a conclusion slide would be good here, Dad. So I'll probably put in one that will say the following. So what we've talked about then is that we've talked about a journey where we've been looking and trying to characterize how a NATO3 gene, or a gene that looked interesting, we were able to screen its potential with using this chick embryonic nervous system model that we have, and then be able to identify that looked interesting, and then take a sort of a risky type of approach to be able to characterize to see whether modifications of that gene could be interesting. And then identified some that were interesting, and now we're putting it out into these human embryonic stem cells to see whether we can actually help drive dopamine neurogenesis. There are some key questions out there, and so no, I'll stop there, actually. So, okay. So I want to thank Mike for his help. The work on the human embryonic stem cells was done during my sabbatical from Grand Valley. And then my other collaborators, some of the work is shown here, some it's not, but Nassim Ben-Ari at Hebrew University, Patrick Brendan at the Van Andel Research Institute, Jeffrey Kordauer, Rush University. And then the support that we've had from multiple different sort of internal and external sources, NSF and others. The students, some of the photos that you saw there were some of the students that have actually worked on this. Dan Doyle is now at the here in the neuroscience program here, and but all of these students contributed and have done wonderful things after they've graduated. Currently, we have Dane Martinez in the lab, who is going to UPenn for an MD-PhD group, MD-PhD program, as well as Melina and Maxwell, who are currently in the lab right now. So, with that, I'll take any questions that you have. Okay, so Dad, common questions that they're going to have are something like, maybe they'll ask about the specifics of the protocol that I have. This is a commercial version of it. And we're going to track at multiple time points here what happens. They may ask some background in terms of other factors that might be important for helping to generate human embryonic stem cells. So I just have some of the papers here that were used to help kind of characterize this. Using LMX1A is one that's been used before, and that could potentially be a gene that could be helpful for helping drive this. And then some, perhaps using this conversion of fibroblasts into dopamine neurons. And so here they found that ASCL1, rather than neurogenin 2, was helpful, and then neurogenin 2 didn't necessarily help. So it could be that this other proneural gene that's a cousin to neurogenin 2, or that was what we used in the human embryonic stem cells, may actually not really be the thing that we need, and maybe we need ASCL1. So so there's some kinds of ideas there. I'm not going to go through these unless there's a question that kind of pulls it up, but that's what I have. If we're looking at signaling pathways that could potentially be involved, so, how does this work? How do you think this actually works? And is this really phosphorylated? And the answer is basically that we're working on trying to characterize the phosphorylation status of NATO3 in differentiation, and looking to see whether it can be actually a substrate for phosphorylation using this mouse midbrain cell line. So those studies are underway now, and would be a nice addition to our work that we're doing now. No matter what, it could be that this PM NATO3 is really important for, we may have stumbled into something that's not necessarily physiologically reflecting a physiological state of NATO3, but still may be able to actually serve to drive a therapeutically useful type of response from neural stem cells. So, more to come. If I had to guess, I have a rather elaborate hypothesis here. And this shows some of the markers that we talked about: sonic hedgehog, FOXA2, LMX1A and B. And this is floor plate cells moving on outward to being more mature. And so this track of all of these different genes that are known to help drive one another. NATO3 in one state could be helpful for keeping a floor plate-like fate and not becoming pro-dopaminergic. And there's a couple, and there's evidence that kind of puts it in this kind of this context. And so what could happen is that in its second state, which could be maybe phosphorylation or dephosphorylation, depending on which which way this actually works. In the second state could actually be that we're actually driving this dopamine neurogenesis that occurs. So we'll kind of see how these different pieces work together. And perhaps it is that, the phosphorylation status of, let's say, S140D is really important for helping to kind of create and drive some of these early types of markers here. And then the dephospho state is the one that actually allows it to release it and to become fully differentiated. Because, when we think about it, doxycycline was kicked on and it was in this stage here. But once we released the doxycycline, that's when we saw the uptick in tyrosine hydroxylase and so forth. So it might have been that kind of reprogramming that was needed. So those are some of the types of questions that might come out. And I think kind of going through here, there's some things I'll clean up. This doesn't quite line up. Doxycycline's added here. This gray is supposed to represent it. I'll go a little slower. I'm at about 46 minutes. So I've got a little bit of time to unpack it, I think. But that's where we are. So I hope that was interesting. All right. What do you think, Patrick? I think it's time to eat.